Institute of Entomology. And I was looking forward to my first presentation in person, but now we have to move to online format. So today I'm going to be showing you the updates on my PhD, and I focus on induced plant defenses, their specificity and roles in plant protection. And I work with willows. So my supervisors are Martin Wolf and Leonardo Jorge. A little background on my research. So the interactions between plants and insect herbivores are primarily mediated by plant metabolites, which can reach astonishing diversity themselves. And studies have shown that this plant chemical diversity mediates not only the reciprocal interactions between plants and insect herbivores, but is also one of the primary sources of their mutual richness. So plants and herbivores have co-evolved and learn how to cope with each other's defenses. And when it comes to plant defenses, we can mainly categorize these as constitutive or induced defenses. So constitutive plant defenses are those that are always present and they are mostly physical barriers, and induced defenses, on the other hand, are those that are upregulated under herbivore attack, and they will cause chemical changes mostly in the plant. So we can also categorize plant defenses as direct and indirect defenses. The direct defenses are those that will have a local effect on the herbivore. So they will be toxic or deterrent or will turn the plant to be non-palatable to the herbivore. While the indirect defenses are extrafluoronectaries or volatile organic compounds that can serve as attractants to natural enemies of the herbivore as predators and parasitoids. But we can see here that to attract predators and parasitoids, the plant needs to show some level of specificity to attract the proper natural enemy of the herbivore. So how can the plant recognize its herbivore to attract a proper response, to emit a proper response? So mostly by the mode of feeding, the herbivores can cause distinctive damage in the plant. So we have, let's say, different feeding guilds. So leaf chewers will cause different damage. That is the damage observed from sap-sucking herbivores, for example, and even between herbivore orders. So caterpillars and beetles and hemipterans will emit different responses in the plant. And also, while feeding, the herbivores left behind little chemical cues in their saliva. So this can be used as a trait that the plant uses to recognize the herbivore. So we can say that insect feeding largely modify plant volatile profiles and causes the induction of blends associated with tissue damage, and also that this production of volatiles can guide insect predators towards their prey. And usually these responses from the plant, these compounds emitted from the plant, are better detectable by the predators than the cues released by the herbivore itself. So this is a very specific host herbivore system, and if the plant manages to succeed and the predator arrives in the host plant, they'll probably suppress insect population and help to reduce plant damage. So within my PhD student studies, I focus on studying how this specificity in induced defenses can benefit the plants. And to do so, I use the genus Salix as my study model. Why I choose willows? Because they are a species-rich genera, especially in Europe, and they have various defensive mechanisms. And also, importantly, they host diverse communities of herbivores. So because of all these characteristics, willows are an excellent model for the study of defensive traits in interaction with insect assemblages. But also herbivores on willows learn how to cope with these defenses, because willows can defend themselves very well, but also herbivores. So these these responses to their hosts largely shapes insect community structure. And willows produce several secondary metabolites as tannin, flavonoids, but also salicylates. And these phenolic glycosides are very characteristic from the Salicaceae family, in which the genus Salix belongs. So salicylates can be very effective against generalist herbivores because they will increase larval mortality and decrease feeding efficiency. But when it comes to specialist herbivores, these defenses are not so much effective, because these specialized herbivores can even prefer to feed on willows that are rich in salicylate, because they can use salicylate as energy source or even sequester this compound to use it as a precursor to produce salicylaldehyde, which can be then used to repel invertebrate predators. And in this sense, willows also produce several terpenes that can be used to attract predators. And this is it's particularly important in the case of suppressing specialized herbivores feeding on the plants, because these other defenses usually are not that effective in these herbivores. So we have another classes of compounds that are being emitted and attracting these predators, as mentioned before. So I have three main chapters within my PhD. The first one focuses in exploring insect traits and how they govern the induction of chemical defenses in willows. The second one focuses in the specificity in these induced defenses and comparing these results among willow species. And as the third module, I will infer how this specificity in induction affect predation in natural habitats. So for this first module, we have herbivore trait, as I mentioned, as driving these responses in the crack willow. And we are inferring this trait, this interaction, using Salix fragilis as our study model here. So this experiment was performed in 2020, and there we use this crack willow, which is a common willow species with diverse chemistry and harbors diverse assemblages of herbivorous insects. And we expose these plants to herbivores in a greenhouse experiment. So we had three main hypotheses, and the first one stated that higher specificity will be observed in VOC production when compared to with metabolites. And why? Because it matters the most to be specific when it comes to VOCs, because this way plants can have higher efficiency in their defenses. And this is very important to attract the proper predator or parasitoid. Second hypothesis was that most of the variation in plant defenses will be coming from herbivore orders or feeding guilds. Because as I said before, the mode of wounding of different herbivores should impact plant responses the most to trigger the production of specific compounds. And also we infer that generalist versus specialist insects will have differential responses. So specifically, we expected that generalists will be eliciting VOCs and with metabolites more or less equally, because they are usually vulnerable to both, while specialists will induce stronger responses in VOCs, because usually VOCs are more efficient against them than the leaf chemistry. So as our setup, as I say, we performed this experiment in a greenhouse experiment. We have 149 cuttings of Salix fragilis, and we exposed them to 24 species of herbivores. So we sample the plants from natural populations, we grow them in the greenhouse, and then we place different insect treatments in each of the plants. We enclose them with tissue bags, and after herbivore exposure, after letting the herbivore feed, we removed the herbivores under frass, and we place the PDMS tubes to sample the VOCs. So we sample the VOCs for 24 hours, and then we remove the PDMS tubes and send them to Leipzig to be analyzed at IDIC. And we also let the plants in the greenhouse for another 6 days, because these other types of defenses, the leaf chemistry, usually takes longer to accumulate. So to be able to properly measure these defenses, the plants were let in the greenhouse for this time period, and then we took in each individual plant and we harvest all the leaves that were inside the bags where we place the treatments. And we photograph every sample, every replicate, every plant, and we measure the herbivory damage of every and each leaf inside the bag. And then we took 3 of these leaves and we freeze dry them and homogenize, and we send the leaf tissue both to Texas to metabolomic analysis and both to Finland to perform tannin quantification. So now I'm showing you the results. As I mentioned, we had 2 different sets of data. So at first, I'm showing results for VOCs for this first experiment. So we quantify VOCs. We were able to find 37 individual compounds. These belong into 5 classes, mainly sesqui and monoterpenes. And as you can see here in the ordination plot legend, we have VOCs as arrows, and each color represents 1 classes of the compounds we found. So we perform a redundancy analysis with stepwise forward selection, and we use the individual VOCs, which are represented here by the arrows, as response variable, and then we use the explanatory ones: herbivore order, feeding guild, specialization, and also the herbivory damage. So when testing for the effect of all these traits and herbivory damage, trying to explain the variation observed in individual VOCs and overall VOC variation, the model selected these two orders as the ones being, at first, herbivory damage, but as you can see here in the urination, herbivory damage plays a very important role, but also here, in the case of VOCs, herbivore identity matters. So not only the damage that's being caused is eliciting VOCs, but also the type of damage and from each herbivore it's coming from. And we can see that there is also a differentiation between beetles and caterpillars. So beetles are eliciting a much stronger response, and they're strongly correlated with several compounds here, especially mono and sesquiterpenes. And then, based on that, we decided to further test the effect of species, because we can see that there is effect from the herbivore. We can see that the herbivore traits are correlated to the overall VOC distribution composition, but also we wanted to see if there is an effect within each order. So we performed separate analysis for both Coleoptera and Lepidoptera, and we perform a partial RDA with stepwise force selection again, and we use individual VOCs, also represented as arrows. And here we use herbivore species. So I'm showing here results for Coleoptera, and we have these species we used for our experiments. And when comparing all these species here, these three were selected as the ones most correlated with the distribution of VOCs. And in this case, we use herbivory damage as our covariate, but not included as explanatory variables, because yes, we can see that there's a very strong effect from the damage itself. But here we were interested mostly in focusing on herbivore identity to see this differentiation. So these three species, you can see there is Chrysomela cupri and Plagiodata versicolora. They are two specialized beetles, and they are eliciting the strongest responses, while Phylogis vidigicalis, which is a generalist beetle, is not so strongly correlated with any of the compounds. But also we can see that there is differences when comparing these two species, Chrysomela and Plagiodata, because Chrysomela is eliciting mostly, let's say, green leaf volatiles, as you can see here. This is methylbenzoate, and the induction of this compound is almost exclusively from Chrysomela cupri. But Plagiodata versicolora, on the other hand, is mostly correlated with the emission of sesqui and monoterpenes. So to further illustrate these changes among herbivore orders, I can show you here the trends in VOC emissions. So when comparing and contrasting this information, we can see that again, all the orders are showing more or less some differentiations, but Coleoptera and Lepidoptera have the higher emissions. And before the dotted line, we have downregulated compounds, and after we have upregulated compounds. And when you compare even the downregulated compounds, you can see that Coleoptera beetles are downregulating only one sesquiterpene, while the other orders are downregulating more than one, and also more than one class of compounds. So the strongest induction is for sure coming from Coleoptera when it accounts for herbivore identity. And now I'm showing you the results for leaf metabolites for this first experiment. So we perform untargeted metabolomic analysis, and we are able to retrieve 42 library matches of identified compounds. So we have 42 compounds here. We have 6 classes across these 42 compounds and plus unknown compounds. So we have organooxygens, organic acids, lipids, flavonoids, benzenoids, and phenylpropanoids. And we perform redundancy analysis with stepwise forward selection. We use individual metabolites in this case, but again, we use the same explanatory variables as before, because we are interested in seeing herbivore traits here and what role do they play in these changes in ingestibility. So first thing we can see is that, again, herbivory damage matters, but here we have several or most of the compounds being downregulated, while we have Coleoptera also selected, but only emitting just a few compounds, and there's like 2 or 3 organooxygens, as you can see here, and these 2 are phenylglycosides compounds, so we know that they play a role in herbivory damage, post-defense from herbivory damage. But of course here, this effect of the beetles is not so much pronounced as the herbivory damage itself. And when we look at the trends, we can see clearly this downregulation, because again, before the dotted line, we have downregulated compounds, after we have new, and there is some patterns for every order analyzed, but Coleoptera is playing it's inducing most of the compounds when it comes to upregulation, downregulation, upregulation. You can see here that there's more classes than the others being upregulated in this rate here for Coleoptera, and also in downregulation, there's some differentiation too. So we can conclude that yes, VOCs show a higher specificity in responses when compared to leaf metabolites, and that changes upon herbivory are driven mainly by herbivore order. And also we saw differences between species within the orders. I didn't show any results for Lepidoptera because we only found marginally significant effects. So I'm only showing for Lepidoptera, for Coleopteras here. And herbivory guild and specialization did not play a role. You might remember that I mentioned that we also performed tannin quantification here, but I also now showing these results because they were not significant as well. So moving forward to further measure these changes of induced defenses upon herbivory, we need to compare the strengths among willow species, because this divergence in defenses can help willows avoid sharing herbivores, and in this way, if which willow species can tailor its unique strategy, it will probably show more efficiency in their defenses. So in the second model, which has this specificity across 6 willow species and 4 insect herbivores, again in a greenhouse experiment, so the selected herbivores show different life stories, feeding modes, and host preferences. We use 177 plants, and the willow species selected were Salix aurita, Salix cinerea, Salix fragilis, Salix purpurea, Salix viminalis, and Salix triandra. And we had 2 species of Lepidoptera, Limantregis Parinoperoptera brumata, and 1 beetle species, Chrysomela punctata, and 1 hemipteran, Afrophora salicina. We exposed these herbivores in the greenhouse experiment. They fed on the plant, we removed the herbivores, we sampled VOCs, then we sampled leaf metabolites, took photographs, measured herbivory damage, all the same as in the first case. And our hypothesis here were that willow species will show differential responses when it comes to leaf defenses, because this evolution is linked to the preferences of the herbivores. So different herbivores will be showing different responses. So this for the leaf chemistry, but when it comes to VOCs, we expected to see very similar responses, because having divergent responses in terms of VOCs could fail to attract predators and parasitoids. If you are sending too many differential signals, this cannot be very helpful sometimes. So when we look at the results for the VOCs, we first quantify the VOCs. So we have 32 individual compounds belonging to 5 classes plus unknown compounds, again, the same classes, mainly monoterpenes in this case. And interestingly, all the identifying compounds here were also found in the first experiment using just Salix fragilis. So there is an overlap of chemical defenses, but here we also found some new compounds that were able to be identified. So we perform a principal component analysis using individual VOCs, again represented as arrows, color-coded arrows. And we had 6 different treatments. So if you look at the plot, we can see at the diagram, we can see different colors, and these colors represent different willow species, but also different shades within these colors represent treatments coming from the insects. So going from the darker to the lighter shade, we have Afrophora salicina, then Chrysomela punctata, then we have the 2 caterpillar species, Limantria and Operoptera, and the lightest shade is for controls. And also analyzing the variance partitioning for the composition of VOCs in this case, we found that the willow species explain most of the variation, and the herbivore species also matter. And we can see here that there is some pattern when it accounts for the variation coming from the plants, so they are differing somehow, but also herbivore identity matters, but not so much. So looking at the trends, we can see that these species are clearly showing different things. If you look at the ones in the left, you can see at first that they are inducing much higher rates of, these are peak areas for compounds, just accounting for the overall emissions. Keep in mind, and we have the classes here. So Salix aurita, for example, is eliciting strong, upregulating very strongly mono and sesquiterpenes, but also downregulating very strongly aldehydes. And the others are also showing this pattern for monoterpenes at least, but the others, as Salix purpurea, Triandra, and viminalis, are not showing such a strong induction, but they are more or less inducing equally all the compounds. But if you take a look on these, just accounting for plant species identity, you can see that there is differentiation. And when you also account for what is happening according to the different herbivores we use in the experiment, we have Salix aurita and Salix fragilis being mostly elicited by beetle, the beetle species we use, while purpurea is being mostly elicited by beetle and also from the Operoptera brumata herbivory, and Triandra and viminalis mostly by the beetle and Limantra gispar, and Salix cinerea being affected by all the herbivores we use. So we can conclude that yes, which species is showing a different pattern in response, and even species that are very similar in most of their traits as aurita and cinerea are showing a very different behavior here. And these changes upon herbivory from different insects is also specific because each of them is doing one separate thing. But again, we can see that the beetle elicitation is the strongest among all willow species, because beetles are the only insect present in all the plant treatments as the herbivore doing most of the damage or eliciting most of the compounds. And okay, now we need to see how to further explore these trends, because we have two experiments answering a lot of questions when it comes to the greenhouse setup. We first test the herbivore traits, then we compare these trends among several willow species, and now we can combine this knowledge from our greenhouse experiments with studies in natural habitats. And why this is important? Because this way we can actually assess and review plant diversity and specificity and see how it's generated and also how affects predation in natural habitats. So for the final chapter, final module of my PhD, we are measuring these dimensions of natural versus induced responses in willows and measuring predation rates, and also how the predation rates are dependent on the herbivore identity. For this, we only use Salix cinerea, the gray willow, and 3 herbivore species for induction, also Pleistocene caterpillars for measuring predation rates. So our hypotheses are that different treatments will elicit different responses in VOCs, because plants can perceive damage by different herbivores and generate specific responses. And also that different treatments will affect predation rates in a different way, because the predators can show particular preferences for a given herbivore and be attracted by different compounds emitted by the plant. So as I say, we are using Salix cinerea, approximately 30 shrubs, 4 branches per shrub. So first branch will have control solutions, which is just mechanical wounding plus saline solution that is also the one using as carrier for preparing the other treatments. Second branch will have mechanical wounding plus treatment from Chrysomela populi extract. Third branch will have mechanical wounding plus Catocala nupta extract. And the final branch will have mechanical wounding plus extract from Notodonta zigzag. And every one of these branches will receive 2 Pleistocene caterpillars. So this experiment will be run for a total of 15 days, and the treatments will be renewed in 3-day intervals. And also, when renewing the treatments, I'll be measuring all the predation rates during this period. So the outputs I expect from this, the first manuscript from the first experiment is going to be submitted in the next weeks. We are aiming for the Journal of Functional Ecology. The second experiments, which were performed last year, also have the manuscript in preparation, but I also waiting for the other results for the chemical data. And we plan to submit this manuscript in later this year in the Journal of Chemical Ecology. And for the final experiment, which will be starting in May this year, we plan to publish next year, or to submit at least next year, in the Ecological Entomology or Journal of Animal Ecology. And I also submitted my literature review, which was focused on the specificity of plant defenses using Salix's study model. I present the results from the first experiment in the conferences last year, and also I'm going to present the results from the first and second experiment later this year. I also did one abroad stay, and I plan to do 2 others this year, and I have a few collaborations with other researchers from our institute and also from foreign institutions. and this will hopefully generate other publications as well. And I would like to thank our collaborators from here and other institutions who are helping through all the steps of this project development. Thank you so much, and thank you for your time, and I'm happy to take any questions.
Okay, thank you very much. It was wonderful. Despite I didn't hear a few bits throughout, I think that you are doing really well in your PhD. So the progress and everything is going seemingly smoothly. And I would like to ask people who have there some questions to specific experiments.
I know we don't have much time, so if any of you is interested in asking anything later, just send me an email and I'll be happy to discuss any further questions too.
Yeah, actually, I don't see whether Ernesto is here already, so.
I am going to mail him again that it will be this room, not my personal Zoom room. So he will join soon. And I have questions for Priscilla. Wait 2 minutes for me, please.
Yeah, go ahead, Olda, with the questions to Priscilla.
I must send a message to Ernesto. Yes, okay. If there is no other question, I would like to ask, in the beginning, Priscilla, you said, or you have written on the slide that salicylates are sequestered by some herbivores as defense against invertebrate predators. Is it right? Did I catch it well? And my question would be, what about vertebrate predators? Are they repelled by salicylates as well as invertebrate predators?
I wouldn't know, actually. Maybe Martin can help me with this because, yeah.
Okay, I don't think anybody has tested that, which will be quite an interesting question maybe for one of future Priscilla's experiments, but salicylaldehyde actually smells really horribly. So even our group members, when we were performing experiments with these chrysomelid larvae, they were complaining about the smell. So it's definitely not very appealing, I think, to vertebrates. So I think that this can also affect vertebrates, but so far as I know, it has been tested against ants and actually lady beetles. These Japanese or like these Far East lady beetles that feed on chrysomelid larvae.
Okay, thank you. I know from my personal experience that the larvae of Chrysomela are really quite bad, and I wouldn't like to eat them. But I had a crazy idea. What about moose? Also, these large herbivores, if they can be repelled from willows infested by larvae of Chrysomela.
Yeah, that would be a very cool experiment. I'm not sure if we can do it here in the Czech Republic, but maybe in Finland or Sweden it could be possible, and it would be really cool to show this, like insect mammalian interaction. But I guess, like, of course, for the moose, it must be pretty terrible to chew on the willow with these chrysomelid larvae on the leaves, because they really smell horribly and it's sticky and it's also very bitter. So the salicylaldehyde is extremely bitter compound, so it's really disgusting in every way.
Yeah, if I can comment to this, because we have been doing the mammal herbivory versus insect herbivory in South Africa with these antelopes. And actually we found that only a few species were actually disgusting for mammals when they were heavily attacked by specific caterpillars. And most of the time they were like disgusting both for mammals and caterpillars at the same time when actually the chemical compounds were active. So I'm not sure how this would work for the moose. I think they would be also deterred if the lady beetles are eating on them a lot. So someone has questions still to Priscilla, or can we move forward to Ernesto?